Hi there everyone, welcome back to the channel. I'm the GCSE science teacher. In today's video, we're going to be learning about DNA structure and the genome, and this is for GCSE biology students. I hope you do find this video helpful, and if you have any questions about it, because lots of the time in real life when I teach this subject, students often have loads of exciting, interesting questions for me. So I'm more than happy to answer any questions in the comment section. Let me know. And if you would like further reading on this and you are very much interested in genetics, just as much as I am, my background is in genetics and molecular biology. So if you are interested in that kind of thing, I will leave some extra reading that's not necessarily for GCSEs, but it's just a bit of fun to help your understanding of the subject further. And I'll leave that in the description box. Anyway, let's get into it. Let's talk about DNA. What actually is it? Well, DNA stands for deoxyribonucleic acid, and this essentially refers to what it's made up of. DNA is essentially the building blocks of life. It's found within the nucleus of our cells, animal cells. It's also found in the nucleus of plant cells, or it can be found free within the cytoplasm in different cells that are non-eukaryotes, so they are classed as prokaryotes like bacteria. And essentially that DNA codes for different proteins. And those proteins could be anything really, from hormones to structural proteins like collagen. They could also be used for receptors on cell surfaces to help with cell signaling, amongst many other things. Even things like in our red blood cells, there is a protein called hemoglobin that helps to carry oxygen around. So essentially, DNA is the OG, the original textbook for everything our body does, but also everything every other living thing does as well. But we are specifically going to focus on looking at DNA structure in a bit of detail, but of course this crosses every single living thing on Earth. Remember, viruses are not really considered living at GCSE. There is some debate about that in the sort of research world, but remember, viruses, they need a host cell to survive. So kind of exclude viruses from this. We don't want to talk about them. They kind of hijack DNA from a host cell and rewire it to make their own proteins. But if we're talking about DNA structure, just remember this is talking about animal cells, plant cells, bacteria. But just remember those animal and plant cells will have the nucleus, whereas bacteria, they do not have a nucleus. It's just within the cytoplasm. But the structure of DNA is exactly the same. So it crosses over everything. So lots of students will often ask when I teach this subject, what is an allele? What's a gene? What's a genome? What's DNA? There's all these different words and they all kind of sound the same. So we do need to kind of refer to this. And one of the questions I've seen a lot in the exam is they'll give you a picture or they'll give you the words and they'll ask you to put into like a hierarchy, which one is the smallest, which one is the biggest, or for labeling purposes, they'll give you the names of these words and they'll ask you to label the diagram. So I've got a picture here that I've drawn for you guys or made for you guys, and this is the sort of thing they could show you in exam. Remember that if we're talking about a cell, it contains the DNA in the cell. So the cell is the biggest thing. And of course, when we have more cells, we have tissues, then we have organs, organ systems, and then the organism itself. But if we go to the single cell, we would have inside of it, if it's eukaryotic, it would have a nucleus. If it's prokaryotic, it would just be in the cytoplasm. But inside the nucleus, we would have what we call chromosomes. Now, chromosomes are these structures of wound up bits of DNA that have proteins attached to them as well. And those chromosomes essentially hold all the information. And in humans, we have 23 pairs of chromosomes in the body cells. But in the gametes, the sex cells, like the sperm and egg, we have just 23 chromosomes there. We have half that number. So in each of those chromosomes, if you were to zoom in on it, you'd see all of that DNA tightly wound up and spiraled in this structure called the double helix. Now, that DNA, if we were to zoom in again, we would have sections of DNA, which is called genes. Now, we're going to look at the structure of DNA in a bit more detail, but I thought I'd mention some of this just to help us understand the next bit of this topic as well. Okay, so let's have a look and see how DNA structure is structured. What does it actually look like if we were to take a piece of DNA and unwind it? What is it made from? So DNA is what we consider a polymer, and you may have heard of this word before in maybe biology or even chemistry GCSE. And a polymer just is a substance that is made up of repeating units called monomers. Think of like a a necklace that has lots of little beads attached to it. Each of those beads are what we call monomers, and the whole necklace itself is the polymer. DNA is exactly the same because it's made up of lots of repeating units. And these units are what we call nucleotides. Now, nucleotides are very important because they have three key elements to them that help essentially generate that code of life. So the first element is the phosphate group, which is found on the outer side of the double strand. So you can see the blue section on that double strand. This is our phosphate backbone, and this essentially is bonded to the deoxyribose sugar, or the pentose sugar. And the reason we call it a pentose sugar is because it has this sort of pentagon shape, the 5-carbon ring structure. And essentially next to that sugar, we have what we call a nitrogenous base. This means it contains some nitrogen. It's just, you can obviously refer to it as a base as well at GCSE, that's fine. And there's 4 different versions of a base. Technically, there's actually 5, but for GCSE sake, there are 4. We have thymine T, we have adenine A, we have guanine G, and cytosine C. Now, all you really need to know about the bases is that they like to bond together in a certain way. And this is what we call complementary base pairing. Now, it's very important that at GCSE you use that phrase, because sometimes I've seen in the past students will think of this like the enzyme lock and key theory, or receptors. And although that's kind of a good idea because it kind of links those ideas together, it's really important you don't use the language associated with like the active site and all of that with enzymes. And that's because we're not talking about enzymes, we're talking about DNA, and they want you to be specific about DNA. So you want to use complementary base pairing. But if I give you an idea of what I mean by that, we will go back to the enzyme idea. If you think about enzymes, they have an active site, which is a very specifically shaped area which a substrate fits into, just like a key will fit into a lock, and it has a very specific, unique shape. Okay, it's the same thing with our bases in DNA. Okay, so thymine, for example, T, can only bind with adenine A. And it's the same with guanine G, only binds with cytosine C. And this just means that the structure of the DNA, when it is copied, because what happens is DNA is found in the nucleus, we want to take that DNA out and copy it so we can make proteins, essentially. However, if you think about like a book, let's think of like a really important book, like a textbook that you're using. If you lend it to your friend, they might forget it. They might be like, oh, sorry, I've left it at home. You don't want it to be lost, or they might write in the book or something, and it changes the story in the book, or it changes the information. That's the same with DNA. So what our cells do, which is really smart, is they will take a copy of that DNA and pass it outside of the nucleus for the ribosomes and all the other cell structures to do what is needed to be done and make those proteins. It's the same with the analogy of the textbook, right? You're going to, instead of giving your friend the whole textbook, you photocopy a page and go, here you go, right? It's the same kind of idea. So that's why those base pairs are there, because essentially once a section of that DNA has been copied, called mRNA, that leaves the nucleus and enters the cell to be used to make those proteins. Now, this whole process, I can do a video on transcription, translation, all of that if you would like more information, but we won't go into huge amounts of detail here because it is a massive topic. But that's what those base pairs are used for. They essentially, the double strand will essentially unwind and unzip, and a copy will be made from one of those strands. So yeah, that's what those base pairs are used for. It's just a way for the DNA to make that double strand and therefore also make copies of itself as well. Okay, so I did want to just point out there is a bit of a story behind the structure of DNA, and this is actually something I'd like to do a bit more of on my channel. I might actually make a series of science stories that links to the curriculum, but also just general science discoveries as well, some of which are very shocking and very interesting. So if you are interested in that kind of thing, definitely let me know in the comment section, and I can make that as a full playlist here on my channel. But essentially, yeah, where did the structure of DNA even come from? How do we know know it's a double helix? Well, it really started off in the 1930s, specifically 1938, where scientists were trying to figure out the structure of DNA. And essentially, some of them started to figure out that it had kind of like a long stranded shape. Some of them referred to it as a cone shape. But in 1951, that's where a lot of the kind of excitement around the structure of DNA started to happen, because in King's College London, there was a PhD student called Rosalind Franklin, which some of you may have heard of before. And her work using the X-ray crystallography apparatus essentially was able to picture there was a double helix shape to our DNA that we now know of. And essentially, the supervisor that she had at the time, the person who was running the research lab called Maurice Wilkins, they didn't really get on very well, as is documented. However, she was his PhD student, so she was doing a lot of the work in the background. And she did essentially come up with the data to prove that it was a double helix. However, Wilkins at the time, like I said, they didn't really get on, and women in science was not as prevalent or as much of a bigger thing as it is now. A lot of it was men academics who would do a lot of the research. Essentially, he would communicate some of his ideas with other scientists. There was some information in 1952 by someone called Pauling and Corey, and they proposed a different model. Watson and Crick also came up with another model that had 3 strands. All of this was disproved as soon as Rosalind Franklin's data came out. And in 1953, Watson and Crick actually used this evidence that Rosalind Franklin had essentially discovered to write a research paper in the journal Nature, which is a massive journal in science, to show this theory of the double helix shape. And essentially resembling this kind of laddered structure. Now, there was a lot of uproar, as you can imagine, later on, because Rosalind Franklin was the person who did most of the work, and Watson and Crick are often the ones who get referred to as the discovery scientists for this particular structure of DNA. However, we now like to credit Rosalind Franklin as well, because of course without her tremendous amount of work and exposure to X-ray radiation as well, because it's a very dangerous thing to be doing, especially back in those days with not much precautions of health and safety. Without that sort of research that she was doing, Watson and Crick really wouldn't have made a big name for themselves. So all these scientists really had their own contribution. Sometimes as well, we call it Watson and Crick base pairing. I know I talked about complementary base pairing before. Sometimes if you read other textbooks, they will talk about Watson and Crick base pairing. But Rosalind Franklin really was the kind of the engine behind a lot of this work. So I thought she was worth a mention. So I will do a whole video on this if you would like me to, but I thought I'd just mention this briefly too. This idea of DNA being used to make proteins through 2-step processes called transcription and translation. Like I said, a whole video is much required on this topic because there's so much going on here. But I just wanted to give you some information anyway, just to help you guys out. But if you do have any questions, please let me know. And if you would like that whole video, also let me know, and I'm very happy to do it. But yeah, protein synthesis. Like I said, proteins can be used all around the body, whether it's a hormone, whether it's a receptor, it could be part of the immune system with antibodies, it could be structural proteins. Proteins are absolutely everywhere. And they come from amino acids. Now, amino acids, there are 20 that are naturally occurring. And these essentially, as they are bundled together from our DNA, they will make a specific sequence. So those sequences of amino acids, that's what determines the structure of the protein and therefore the functionality of the protein as well, and the type of protein that's made. So all of this is based on the DNA structure. Okay, so the structure of DNA, those base pairs that we talked about, A, C, T, and G, the structure of those, whether it's A, T, C, G, A, T, G, whatever it is, the whole sequence, once that's copied, it essentially is used to make the amino acids, and then it's used to make the proteins. So if there are changes to the DNA, it can affect the amino acids that are made and then the proteins. So there's lots of things that can affect the protein that is made. And that's why those different combinations of amino acids, although there's only 20 naturally occurring, because there's so many different combinations we can make, that's why we have so many different types of proteins as well. So it's pretty cool in that way. So what do you need to know? You need to know that proteins are made from DNA, of course, and this happens in 2 steps. So the first step is the transcription step, and the second step is translation. So just to help us visualize this, remember transcription, if you look at the word transcription, it has the word script in it. And I want you to think about a script as a piece of writing that you're reading out loud. So you're copying the words off the page. Now, sometimes mistakes can happen. You'll read something and you'll misspeak or you'll say something incorrectly. And that is exactly what can happen with DNA. It can be misread or it can be misinterpreted. And this can lead to problems later on down the line. Translation is when that bit of information, like in 2 different languages, from English to French or Japanese to German, you're translating it into a different form, but the information is still the same. It's just you're changing the code of it. You're changing the way it's presented. So let's talk about this in a bit more detail, but those 2 phrases often come up in textbooks and things like that when you do your own revision. So it's just worth knowing what they mean. So transcription is talking about copying a piece of DNA into what we call mRNA from the nucleus. And that mRNA will then move out of the nucleus to a ribosome, which is a organelle inside the cytoplasm. Now, at this stage, we have translation. So everything before, just copying that DNA, that's transcription. Translation, like I said, is changing the format of what is being spoken, whether it's English to French, for example, right? What we're doing with translation is we're going from DNA to protein. That's what we're changing something into something else, right? So what we have is we have our mRNA, that copy of DNA, it attaches to the ribosome, and that ribosome will actually read the base pairs, so A, C, T, G, all of those base pairs, but it will read it in threes. And this is what we call a codon or a triplet code. And 3 bases will code for 1 amino acid. So you can see here in the diagram, we have ATC, that's 1 amino acid. AGG is 1 amino acid. CGC or GCG is 1 amino acid. My eyesight's clearly going. That font's quite small. Maybe make it bigger in editing. But essentially, those amino acids are made by reading the ribosome reading along the strand of DNA, and it does that in threes. Okay, so once that's done, those amino acids actually join together, and that's what forms the protein. And that protein can then wind up and form that nice structure, and the structure of it will help it function. So things like an enzyme, for example, the shape of it is super important because of the active site. Right. So those are the sorts of things that you want to link those ideas together. And all of this is really for those higher tier triple scientist students. So if you do have any questions, like I say, do let me know in the comment section. I can definitely do a whole video on this. Okay, so for the most part, this is done, transcription, translation is done amazingly well. Our body is super smart. Other organisms who have the same process of transcription and translation, plants, bacteria, more complicated organisms, of course, transcription translation works amazing. However, because of environmental factors and also because of genetic factors, sometimes our DNA mutates or it changes, and this therefore can change the overall protein that's made. And this can lead to problems. It can lead to diseases. It could lead to proteins not working properly and therefore causing diseases. It could also be silent. It might have absolutely no effect. And it also could actually be beneficial. Sometimes mutations will actually help an organism evolve over time, and those advantageous genes are passed on. So essentially, mutations drive evolution. So that's something to think about as well. But yeah, DNA, the structure can change. This will obviously change the protein that's made. And I just think it's worth mentioning at this stage that it's also environmental, but also genetic factors that do this. So things like carcinogens, we know, can cause cancer. Cancer, of course, is the rapid changing of our DNA and cells, and uncontrollable division of cells. I have done a whole video on cancer if you are interested, so I can link that for you in the cards or somewhere in this video. But yeah, so it's all those ideas that we've talked about before, linking them together and understanding that This is not just a standalone topic. Try and link those ideas together, and that will really help you understand the topic further as well. So last but not least, we're going to talk about the Human Genome Project. And this links nicely to this video because, of course, the title of it is referring to the genome. So what is the genome? The genome is just all the genetics, all the DNA that's found in a certain organism. So it's essentially the genetic makeup of that organism, what makes a human in terms of its genes. It's the same with other organisms as well. We've actually got lots of data on lots of other organisms, and some organisms are actually used more than others for research purposes, like the Drosophila fly, Drosophila melanogaster. That's a really key organism that's used because it's got quite a small genome compared to other ones. And yeah, the human genome was a massive project, essentially, where scientists mapped all the DNA, all the genes, and it took them quite a bit of time. It took them from 1990 to 2003. And essentially, now we've been able to do this. We can do a lot of amazing work as scientists with this information. So, some of the things they want you to know at GCSE is that the Human Genome Project happened at this time, but also it can be used to identify genes linked to different types of diseases, things like cancer, for example. BRCA genes in breast cancer are key examples here. We can also use it to check inherited disorders and check the research and add to this research of different inherited disorders. We can also use it to find new treatment methods for diseases as well, and also discover any risk factors for diseases. So, a lot of the research that we know about cancer, for example, we know about lots of risk factors because of the human genome, and we were able to find this information out and compare different bits of genes from other people who have the disease, who don't have the disease, and find this information out and get essentially a bank of wealthy knowledge to help people with cancer and also prevent people getting cancer as well. We also can use it to track migration patterns from the past. So, of course, everyone around the earth has lots of different characteristics based on their ethnicity, and we can use it, the Human Genome Project, we can actually use our genetics to find out where our ancestors from. You may have actually heard of some of the test kits you can buy. So 23andMe, that's a key one. There are loads of other ones that do this, but essentially you would take like a saliva sample, you send it off to a lab, and it will tell you all your ancestry information. It will tell you where your ancestors have come from around the globe, which is pretty cool. And again, this is all based on the research of the Human Genome Project that we know of. And then lastly, we can also use it in the medical field in terms of genetic counseling. So if somebody is wanting to have a child and they are high risk for certain conditions, maybe things like sickle cell anemia, thalassemia, other different conditions as well, we can actually use the Human Genome Project and the genetics of that individual and the partner to figure out what the likelihood is of that person having a child with that condition. And this links on nicely to a video I'm going to do shortly, coming soon, which is also to do with Punnett squares and inheritance. And that's it from me today. I've been the GCSE science teacher and you have been curious. It's been lovely hearing from you guys, especially over the summer when a lot of you had your GCSE results come out. And also lots of you have been DMing me on Instagram, asking me for revision and, help with the next academic year. So I hope you did find this video helpful. If you haven't already, please do subscribe. If you know of someone that would perhaps enjoy revising or maybe needs a little push into revising, do share this video with them. Feel free to tell them about this channel so we can all build this wonderful community together. So yeah, thank you so much. Have a great day and I'll catch you in the next one. Take care.